Good afternoon, everyone, and welcome to the February 24th Mitochondrial Damage Modeling and Measurement Group meeting. My name is Shane Kennedy, and today I'll be presenting on the topic of measuring cell viability using a combination of Hoechst and Ethidium Homodimer 3. We're going to talk about cell viability and mitochondria and the relationship between the two. I'll be talking a little bit about the MTT assay, just because it is such a common method of measuring viability, even though that's not our main topic today. I'll be discussing cell viability with Hoechst and Ethidium homodimer. I will talk about the background of both of these compounds, the protocol we use to use them, analysis methods, and then I'll open it up to questions. So let's get started. People in our group are involved in measuring mitochondrial damage and modeling it as well. However, mitochondrial damage is one of the many things that can cause apoptosis. There are a number of mechanisms by which this can occur, but it is known that ruptured and damaged mitochondria themselves through cytochrome C release are one of the #1 triggers of apoptosis in the cell. And so we need to know if our damage intervention is actually causing the loss of cells and damage to the mitochondria itself in order to determine what's actually going on. So I'll give you one example of this. There was a 2005 paper by Kujoth et al. That used a mutant mitochondrial DNA polymerase gamma mouse model to determine the relationship between aging and mitochondrial damage. And they had this mutant mouse model, and they found that the mice aged much more quickly and exhibited aging phenotypes much earlier than the control mice. And so they looked at the cells in the mouse and measured mitochondrial damage. They found, however, that there weren't that many cells with mitochondrial damage or mitochondrial mutations in the tissues they measured. And that's very strange. What they did find, however, was that the tissues in the mice had a loss of cells, a loss of cell number overall. And that's the thing that was causing the aging phenotype. So what was happening is that the mitochondrial damage was causing aging, not by causing the mitochondria and the cells to function less well, but by driving the cells into apoptosis, and then the loss of cells was what caused the aging phenotype. And more specifically for our purposes, if we're working with modeling mitochondrial damage in cells in culture, and we induce our damage model, but we find that there is no damage, well, the question is our damage model not working, or is it working so well that it's causing cell loss, and we don't measure the damaged mitochondria and mitochondrial DNA because the cells that contain them become apoptotic and so are no longer around to be measured. So even though what I'm discussing today isn't a measurement of mitochondrial damage directly, it's a measure of one of the very important consequences of it. And as people studying that, we need a good way of ascertaining cell viability. Now, one method of measuring cell viability that you'll see in a lot of papers is the MTT assay. Here's where the acronym comes from. I won't read the compound because it's difficult to read, but very briefly, the MTT compound here is converted to formazan by oxidoreduxases in the cell. And then these crystals, the formazan crystals that result, have a purple color. You can measure this in the spectrophotometer. I think a lot of us have probably done MTT assays. So what is occurring here with the MTT assay is it's actually measuring the reduction power of all of the cells in the well. And one of the biggest components that does this is the electron transport chain. In fact, you can see here at the end of complex 4, oxygen is reduced, thereby acting as an oxidizing agent by the electron transport chain itself. So this is a very powerful engine, the electron transport chain, for reduction. And in fact, it's actually possible to alter MTT readings by adding anti-myosin A to to cells. So what you're actually measuring here is total reduction power in each well. Very typically, people seed their cells in a 96 well plate. They have their control, their treatment group. They incubate the cells with the MTT reagent, add DMSO, dissolve, measure the color via spectrophotometer like you see here. However, this MTT assay has a number of limitations. You're assuming that you have seeded the same number of cells in each well, which you almost certainly didn't. You're also assuming that your treatment, whatever that is not impacting oxidoreductase performance in the cell. So you're assuming that the cells are able to have just as much reducing power under your treatment as they do not under your treatment. You're also assuming that you have added the DMSO and MTT reagent to each well at the same time. Now, you can mitigate this with good pipetting and a multi-channel pipette, but it's still not a reasonable assumption. And here's another interesting assumption. You're also assuming that the density of cells doesn't have an impact on mitochondrial performance. So maybe you in this cell you seed cells at a lower density than this one here. You're assuming that the lower density isn't causing the cells to have more reduction capacity. Now, you can correct for some of these problems statistically, having a large plate, large sample size, measuring standard deviation. And you can also do a kind of a density curve, which is what this person has done here. But you're never going to totally correct for all of these components. And actually, some work that I've done indicates, in our cells at least, that having a lower density of cells actually leads to cells expressing, on a per cell basis, more electron transport chain proteins. So that's a very interesting thing we find. And for this reason, I recommend against relying on MTT assays for just in general, but in particular, if you have something that will modify mitochondrial performance. Because if you have an intervention that damages the mitochondria and inhibits their reduction capacity, and by extension, the total reduction capacity of the cell, that's going to show up on the MTT assay, even if the amount of cells doesn't change. We can solve this problem by staining all of our cells with Hoechst, staining the dead cells with Ethidium Homodimer, counting the cells computationally, and comparing the treatment group to the control. And we can avoid these problems. Let me talk about how this is done. Firstly, total cell number can be measured with a compound called Hoechst. This is a compound that stains all of the nuclei and all DNA in the cells, and by extension in your well or your treatment dish. Now, unlike calcine, which is another way of doing this, Hoechst stains the nuclei in the cell. Calcine, which is a live cell stain, stains the cytoplasm, so it becomes difficult to count individual cells. Now, someone in our lab, Lisa Salmeron, has used calcine and Ethidium Homodimer to measure cell viability in her artificial corneas with a good deal of success. However, it does not allow you to count individual cells. You're just relying on total intensity, and it does not allow you to also know the total number of cells. You're not going to be able to count cells individually, and so you're not going to be able to detect a small change in viability. And this is important, especially for those of us that study aging, because we can't age a person for 70 years to study them. But if we can improve the quality of our assays to where we can reliably measure smaller changes, we can spot an aging associated trend that might be important, maybe not in the short term, but in the long term. So by staining with Hoechst, we can count all cells, live or dead. Now, for staining dead cells, we use Ethidium Homodimer. I think a lot of you may have worked with Ethidium bromide. I know I have. And this compound is a dimer. You can see it here of ethidium bromide. This is ethidium bromide on the left. So it's very simple. And this compound has a positive charge and it can't cross the cell membrane when the cell membrane is intact, so it won't stain live cells. And you can measure this. You can count your cells with flow cytometry, or you can use a fluorescent microscope. And Ethidium homodimer fluoresces on about the same wavelength as Texas red. So it's fairly easy to measure in that sense. Now, Ethidium homodimer, because it is measuring dead cells and cells that might be undergoing apoptosis, the cells that are stained by Ethidium homodimer and also Hoechst can vary in terms of size and morphology. So when you're doing this, you'll want to have clearly defined cutoffs as to what counts as a dead cell versus what counts as maybe a piece of debris. And I'll talk about exactly how this is done a little later. I would also note that Ethidium homodimer, the stain, I think it stains debris continuously with time. The intensity of the stain increases continuously. So you definitely want to wash it afterwards. And that's especially important because you're going to have these objects that get progressively more and more stained with time. So if you leave the Ethidium homodimer in there and you measure your control first and then your treatment, it'll look like your treatment has more dead cells. That's just because the Ethidium homodimer stain is getting stronger with time. So I recommend definitely wash it out before you image so that you don't have that problem. In addition, you're going to need a clear positive control as well that has dead cells and dead nuclei so that you can calibrate your dead cell measurement to that. This is actually a little bit more difficult than we thought it would be, but overall, I found that the most reliable control is a 100% ethanol incubation at 10 minutes as a positive control for dead cells. And that's what you can see here in this image on the right. All of these are dead cells that were treated with 100% ethanol for 10 minutes. And they look a lot like nuclei in live cells. You can see this right above my laser pointer, that little tiny structure there. You get a lot of stuff like that with Ethidium homodimer. And so you need to have a clear cutoff. And by comparing it to these other things here, which are definitely nuclei, you can set a clear cutoff for what counts as a dead nucleus or the nucleus in a dead cell and what doesn't. So with these two compounds available, here's the protocol that I have found works relatively effectively. You'll perform your dead cell positive control with 100% ethanol if you want first. Then you'll rinse that out. You'll incubate all of your cells with Hoechst for 10 minutes. Rinse. Add Ethidium homodimer 3 for 30 minutes. Rinse. Add PBS. And then image on a fluorescent microscope. And you can see that we get an image something like this. This is Hoechst. And you're going to have two channels. You're going to have the Hoechst channel. You're going to have the Ethidium homodimer channel, which is going to be the same as Texas red. Now, here's where I think this is very helpful. Once you have these images, it's possible to count individual nuclei. And we can do this with a custom MATLAB script that we've written. And you can see a demonstration of it here applied to a Hoechst image. So what our script will do is it will count structures and it will determine the number of nuclei in each structure. This is a relatively crude and unoptimized version of the script. It doesn't get it right all the time. So you can see here that I think is probably 2 nuclei, but it's labeled as 3. That's 1, but it's labeled as 2. But the great thing about using this method is that you can actually customize exactly what you want the script to count as a nucleus versus debris. So you might in our case, you could say any structure that's round, like this one here, counts as 1 nucleus. Anything that's kind of oblong counts as more than 1. You could set a minimum threshold. These cells in particular tend to have nuclei that are kind of oblong shaped. These are TM1s, and you can see that's what's going on here. So you can modify it to count whatever you want. And the thing I really like about this compared to MTT or compared to calcine is that you get a total hard integer number of the total cells. That's what Hoechst is staining. and the dead cells as well in Ethidium homodimer. And you can do all kinds of quality control when you analyze with this script, which I will make available to anyone who requests it. And you can really see exactly what's going on, and you can eliminate the ambiguity. You can get a very precise measurement of cell viability, and then that will allow you to reliably detect even small changes in viability that occur as a result of your intervention. So I would like to now open the presentation up to questions if anyone has any.